Hi everyone, my name is Jaime Brambila. I'm the co-founder of Grace Health and Wellness. My other co-founder is my wife Nicole, and she is the subject of the case study that we're going to be presenting today. And my wife Nicole is right here, joining us today. So the title of the case study presentation that I'm making is Metastatic Breast Cancer in Remission Following Standard Cancer Treatments and Adjunctive Medical Cannabis and Psychedelics. So in August of 2018, Nicole was diagnosed with stage 4 de novo metastatic breast cancer, which was an ER positive, PR negative, HER2 positive, BRCA negative, ductal carcinoma. It's a mouthful right there. So it included metastases of the bone, liver, and lymph nodes. When we first spoke to her oncologist at the clinic at Memorial Sloan Kettering, her prognosis was less than favorable. They explained that she had to go through many rounds of chemo, radiation, and perhaps, a life that would be filled with therapy for the rest of her life. So here we can see the scans, the PET-CT scans of when she was initially diagnosed. I don't know if you can see my cursor there, but so if you see the slide on the left-hand side, that's the initial mass, right there, that's the breast mass. And then on the left-hand side, we see that these are metastases of the pelvic area. So this was when she was first diagnosed. She took standard therapies in combination with the cannabis and psilocybin. And so the chemo and targeted therapy regimen that she underwent was a combination of Taxotere, Perjeta, and Herceptin. And in the oncology world, they call this, they studied this through the Cleopatra study. So what we know from that study is that 37% of patients are alive at 8 years of age. When a patient becomes metastatic, their lifespan is dramatically decreased. Some statistics show that at best, in the best case scenario, patients get to live up to 10 years if the treatments are successful. It's a life of constantly changing treatment because the cancer becomes resistant to the therapies, and so oncologists have to constantly switch the therapies. So only 16% of patients with this diagnosis and with this type of protocol experience no evidence of disease. And that's very important because patients that do experience no evidence of disease, that's an indicator of how prolonged their lifespan can be afterwards. In addition to this, we did do a ketogenic diet because we had the understanding that cannabinoids are lipid molecules, and so if we optimize the lipid metabolism, there's a better chance of absorption. We also included supplements, borage oil, omega oils, all precursors for endocannabinoids. One thing that I want to point out here is the PEA. Let me go back. So the PEA is an important supplement because that's an endocannabinoid mediator. And so there's limited studies on how this helps absorb cannabinoids, but we believe that it potentiates the effect on the absorption rate of cannabinoids. So obviously we knew when Nicole was diagnosed that cannabis would hold some sort of answer for her. We didn't know what that answer would be. At best, we thought that it could help with her standard therapies because standard therapies have very, very harsh side effects that regular pharmaceuticals are not able to control. So you have patients that have severe vomiting, severe anxiety, severe pain. And the conventional tools that oncologists have are not that effective, even though they want to convince us of that. CBD really offers a much better response to mitigate those side effects. So this was the protocol that we figured out for Nicole. And if you see, this is the, sorry about that. This is the collective result of all the certificate of analysis that we did on 7 different extracts. Those range from high CBD cultivars to high THC cultivars, some 1-to-1. And you'll see that she was taking some pretty heavy doses of THC over here. Obviously, we titrated slowly to desensitize her to the motor impairing effects that cannabinoids have, and we can thank our mentor, Dr. Dustin Sulak, for providing that protocol. So one thing that I want to point out with this is that if patients are seeking to use cannabis in their cancer treatments, if you see the amounts that Nicole was taking, you can't just go buy this at a dispensary. So in order for us to create these medicines and make sure that they were the highest quality, first of all, we had to build a lab. We had to understand how to extract using sophisticated methods to really extract the right therapeutic compounds without compromising the acidity in the compounds. And then we had to seek knowledge from practitioners that have many years of experience to figure out how to effectively dose these formulations. So very important to take note, because one of the points that I want to make here today is that even though we're going to go through all this beautiful and wonderful information, we don't have this available in the clinics, even though we really should. So cannabinoids have many different mechanisms of action to turn off a cancer cell. And so the chart that you guys see over here is just a summary of all these mechanisms that take place when cannabinoids get to the cancer cell and exercise that anti-neoplastic effect. So we know that cannabinoids are amongst the most potent adaptogens in nature. So when we take phytocannabinoids, we strengthen our endocannabinoid tone, which produces homeostasis across all of our physiology. So these compounds have been found in preclinical studies to work synergistically with, specifically with HER2-targeted therapies. We know that independently, cannabinoids can turn off ERBB2-driven breast cancer cells, which are HER2-positive breast cancer cells. And then, obviously, we know that cannabinoids have very, very strong palliative effects so that we can mitigate those side effects from standard therapies. In addition to this, I'm just going to go through some of the main mechanisms of action. So cannabinoids have been found to inhibit AKT-mTOR axis. This is a very important, this is an important target in cancer drug development because it induces apoptosis when it's inhibited. So cancer drug developers have been trying to figure out how to turn off this pathway and have not been able to do it effectively without harsh side effects because of what it does to healthy cells. In addition to turning off AKT-mTOR, cannabinoids also activate mitogen-activated protein kinase, so MAPK signaling pathways are involved in proliferation, differentiation, and invasion of cancer. And also we know that, now we have 2 main targets that cannabinoids can effectively bind to and can turn off a cancer cell through. So we also have nuclear factor kappa beta. This is a very important target in cancer development because this allows the epithelial to mesenchymal transition. So for those that are not familiar with all these terms, basically that's when a cancer cell turns from an epithelial cell to a cell that can actually move throughout the body and can attach so that it becomes invasive. So CBD promotes DNA binding of NFKB, nuclear factor kappa. And this has been found in glioblastoma multiforme. So glioblastoma cells, glioblastoma cancer stem cells actually are very, very hard to target. We have some targeted therapies out there that, well, I guess I'm not an oncologist to say that they're effective or not, but the success rate with those targeted therapies is not very high. Cannabinoids can effectively target this and can halt the DNA transcription of cancer cells. So another very important target are PPAR receptors, and these are nuclear factor receptors. In particular, PPAR gamma is very important to our metabolism because that's the factor that is in charge of differentiating adipose tissue. So we know that drug developers are targeting these nuclear receptors because they're finding out that if they influence these receptors, cancer cells are being turned off through different targets that create apoptosis. So when cannabinoids bind to these receptors, these are activated, and they have been found to create what's called lipoapoptosis. So there's a buildup of lipoproteins inside the cell, which creates the apoptosis, and cannabinoids are able to stimulate this as well. So through the hallmarks of cancer, we have cell differentiation and progression. Then we have angiogenesis, which is when a tumor becomes vascularized. So tumor sends the signal to the brain that it needs blood vessels to feed off of. That happens through the creation of HIF-1. And so cannabinoids have been found to inhibit HIF-1, which prevents angiogenesis in lab studies. And we have the last hallmark of cancer, which is metastases. Once the cancer becomes invasive and it attaches to another area of the body, it starts to build another tumor mass. Another very, very important gene target in that process is called ID1. And cannabinoids have been found to block ID1. So in addition to cannabis, we also use psilocybin therapy. And initially when we knew that psilocybin was going to work, we didn't know that it had immunomodulatory properties. We were just using it because we wanted to mitigate the depression that a patient goes through when they're going through cancer treatments and when they're diagnosed with a terminal cancer diagnosis. So we selected 3 types of strains, psilocybin galandoi, psilocybin tamponensis, and golden teacher. All of them were grown by me. So we did large dose sessions, and what I did for Nicole was I steeped the mushrooms, stems, caps, and truffles in boiling water to separate all the therapeutic compounds from the mushroom fibers. The setting that we used was in a safe, harmonious environment with dim lights, minimal sound. There was a mask placed on our eyes to direct the experience inward, because that's where we find that the emotional release happens. There was a guide present to make sure that she was safe throughout the session. And then the day after, there was a reintegration counseling session that takes place. And this is so that the patient can identify the emotional release that happens during the session. So she was dosed 4 grams in each session. One took place in November of 2018, the other one in January of 2018. And then she also did intermittent microdosing. So psychedelics, tryptamines in particular, also offer potentially anti-cancer mechanisms of action. So we know that psychedelics stimulate 5-HT receptors. When 5-HT receptors are activated, 5-HT2A in particular, which is the psychedelic receptor, these create heterodimerization. And basically what that means is that it's crosstalk between receptors. When the serotonin receptors activate, it crosstalks with the MAPK receptors, and it activates a downstream signaling cascade, which eventually inhibits PA3 AKT pathways. So also, these compounds influence nuclear factor kappa beta. And if you notice, I'm talking about the same targets that cannabinoids can influence. So there's a theory that using both compounds, we can potentiate the effect, and that would be something very important to research in the clinic. Also, we know that psilocybin inhibits HIF1, which is that factor that I was explaining is crucial in angiogenesis. So what were the results of Nicole's treatment? So if you see the slides on the left-hand side, you see the initial slides. 5 months after treatment, only 3 chemo rounds applied, they found that Nicole had no evidence of disease. And that was a miracle that we experienced. We didn't know that this could happen. And we didn't know if the cannabis that we were using was gonna be effective. We didn't know how the psychedelics were gonna influence her health. But this is what we received. So how do we prepare these medicines so that we can have pharmaceutical grade cannabis? Well, it's an intricate process. At the crux of this, we have the issue of sustainable reproducibility. The chemotype expression, as probably the presenters before me explained, is very hard to achieve because of the heterogeneity of cannabis and how it produces that chemotype expression. So in order to pull this off, we need some sort of genetic sequencing to lock that genetic profile. We need controlled environment so that the plants only produce those chemotypes with the environmental setting that is controlled so that it can produce that. We had to use standardized extraction method. So we used cryogenically treated ethanol extractions, and we prefer that because it's been established that it's a reliable method so that you can extract the acidic compounds without compromising them. So we extract at very, very low temperatures, -60 Celsius actually. So German Pharmacopoeia has identified this method as the method of choice for cannabis extractions. And handling the acidic compounds becomes very critical because they're very, very sensitive. So any temperature change, even throughout metabolization, we don't know what happens to those compounds getting to the cancer site. To minimize any margin of error, we had to enlist the help of very knowledgeable scientists that were able to produce mass spectrometry analysis on our extracts, so we know 100% of the content in our chemotype expression. So why whole plant and not isolated cannabinoids? Well, we know that whole plant extracts create, we have the theory that it creates the entourage effect. Most studies have been done with isolated cannabinoids. Study at Technion showed that pure THC did not produce the same effects as whole plant extracts. We have a paper that came out of Guzman's group in Spain that showed efficacy between isolated cannabinoids and whole plant extractions. And in particular, whole plant extractions were effective at turning off luminal B or ER-positive, PR-negative HER2 cancer cells, and triple negative, which is triple negative is still remains the most difficult to treat breast cancer. So for patients suffering with these diagnoses, this becomes very critical for them. So essentially what we're advocating for is so that these compounds become part of the armamentarium to treat breast cancers. So what can we draw from understanding all of this knowledge? Well, we know that if this was to get into the clinics, it would do a world of good for cancer patients suffering through this. We know that late-stage oncology treatments are limited. We know that patients create resistance, and oncologists have to constantly try to change therapies. So we have increased number of people that understand these statistics and are opting to include cannabis and psilocybin in their treatment regimens. And recent systematic reviews have validated that we have tons of preclinical evidence that cannabinoids can produce these anti-neoplastic effects, but yet we don't have relevant research. We know that there's a lot of real-world evidence out there, and we see physicians and scientists that understand this effect trying to get this published into the literature so that they can show physicians that this is real medicine. You know, we're not pulling the wool over anybody's eyes. And if we can just get rid of that stigma that people have with cannabis, this could really be helpful for people. So I want to just give some examples of the real-world evidence that's published in the literature. Here we see the first pilot study that was done in Spain with patients that had terminal glioblastoma multiforme. We had very, very promising data from that clinical trial. We see that there are other case studies that are being published, individual case studies of patients that are experiencing this, taking cannabinoids. So we have small biotechs all over the world that are trying to figure this out and figure out how to take these compounds through the drug approval process. Here is an example of a company out of Israel that claims to have produced a formulation, a cannabis formulation in combination with mushrooms that is effective at treating breast cancer. And where are we and how do we fit into everything that I just explained? Well, you know, as I explained before, receiving this gift that we regard as a miracle is a powerful thing to absorb. And we can't really help to think that it was for a very important reason. And that reason is to figure out how to get this into the clinics, how to really deliver this into patients' hands. And, you know, I really just want to make a note of this. We really enjoy the benefits of legalization, and that's great that we have dispensaries in our locality, but true legalization, true access is an illusion because it's limited by the budget that people have to buy these medicines in a dispensary. And if you have walked into a dispensary, this can become very, very expensive very quickly. If we were to buy these medications in a dispensary, these medicines, we would be spending about $15,000 every month, which for a regular person, how can you ask them to make this expense to save their life? It's impossible. So what we've done is we've reached out to people that really identify with what we're trying to do. A group of researchers from Imperial College of London, researchers from the University of Connecticut, and we have put together a clinical pipeline so that we can test our proprietary extracts and in vitro models so that we can screen for efficacy of the anti-cancer effect that we're seeing in real-world evidence. We are going to start our preclinical studies sometime in October, November. We hope to come back next year to give you the results of those studies. And so we encourage everybody to look into this, really. My wife Nicole's testimony that this medicine is a powerful medicine, and she's just going to come up stage really quick and just deliver a message for you guys.
Thank you. I'm really bad at this, so please be patient. What I just wanted to also add to what Jaime has been explaining is the following. Before I begin, Jaime and I want to thank the many pioneers who began this revolution and who started before I was even a blip on the radar screen. If it weren't for all of you, none of us would be here. You are the heroes who fought for this miraculous plant, who saved so many. We're grateful for your advocacy and are blessed to be a part of the revolution in medicine that's occurring. Cannabis saved my life. It's that simple. In 2018, when I was diagnosed, when I was told I was probably going to die, pretty soon, the first thought that entered my mind was, what am I going to tell my mother? That was the first. And then the second was, damn, I certainly don't have enough money to buy the Ferrari I wanted. So we're going to have to work on that. I was afraid of everything that was about to occur. I was a high school English teacher, and I was the one who was telling my students, you know, be careful what you're doing. Cannabis is not maybe the greatest thing, especially since you're not coming to class. And that's because of the stigma, which is the really unfortunate part. So this has been a fantastic lesson. Because of the negative connotations with which I was indoctrinated regarding cannabis, my knowledge as to the benefits were totally unwarranted, untrue, and ridiculous. When my treatment began, I was ready, and I had a cannabis arsenal behind me. And of course, a husband who was willing to sacrifice everything to save my life, and that was pretty cool. I was able to sleep, I was able to eat, and I was able to function, even after 8-hour spinal surgery as a result of the cancer eating away at my 4, 5, and 6 vertebrae. Doctors told me that I wasn't going to be able to move for probably about 3 months. After about a month and a half, I was up, out, working, driving without a problem. There were minimal side effects, minimal. And what's very interesting is most of the medications, as Jaime was explaining, are so incredibly debilitating that the only thing you want to do is crawl up into a hole and die. And sometimes it's the medications that make it worse than the actual cancer. And that needs to change dramatically, because there is absolutely no reason for suffering at this point with what we know. So my question to all of you, all right, how many of you have been affected in some way by cancer? Family members, friends, yourselves. Okay. How many of you have been with those people while they're recovering from chemo, from radiation, from just the depths of hell with what those medications do? And at this point in time, if we have that knowledge, how dare us not alleviate that suffering for everybody else? So for this, I don't want this to be in vain. We want to pay this forward and stop the suffering. And that is what is at the root and the crux of everything that we do. And that's why we're here today. So I thank you for your support, and I thank you for the research and for everything that you're doing, because this is the change. We're making history. This is where it starts. So thank you.
Thank you, everybody.